So let's go in the proof of principle. So to illustrate you a bit the power of chemical genetics, I need to go back at the beginning of my career. So at the beginning of my career, I was working in Sanofi Aventis in a pharmaceutical company in Europe. And the idea was to set up large-scale screen, to set up high-throughput screening to find drugs, in vitro high-throughput screening, to find drugs that would bind or modify the activity of a specific target, a target known to be involved in a disease. At that time was Alzheimer, Parkinson, and a few others. And then finding such compounds, we were trying to climb the ladder of a traditional drug discovery pipeline to find, to develop a drug for human. And they were involved in different departments. By looking at that pipeline, that traditional drug discovery pipeline, you may understand that if you don't have a good target to start with, it's very hard to develop a good drugs for human. And if you don't have target, and unfortunately, most neurological disease and neuromuscular disease, the disease I'm working on, are poorly understood. We don't understand them well, so we don't have good targets, and even for some, we don't have targets at all. So this whole pipeline is not working well. And if we take the example of MND, of motor neuron disease, it's even worse because these diseases are considered as not bankable, meaning the industry doesn't feel that these diseases are value for money for them. So they don't put a lot of effort in trying to identify targets and establish these screens. They wait us, the academic system, to bring these targets on the table to then initiate the screen or to bring compounds already developed. So yeah, so this pipeline is kind of useless until you don't have a good knowledge of your disease. So at that time, I was then very attracted by another approach which was emerging in the academy, in the university, in the public system, sorry, which was the idea was to use small animal models and phenotypic screening. Meaning trying to reproduce your disease in animals such as sea lions and zebrafish that are compatible with large-scale screen, and try to get a phenotype that is relevant to human. And then, instead of looking at a target, you look at the phenotype, and you look for compounds that reverse this phenotype, all right? And so that's what my idea. I wanted to go in the university to do a PhD to demonstrate that it was indeed feasible and try after to sell that to the industry at the World Department. But I was also at the same time very attracted by basic research, by actually understanding what I was doing, understanding the disease I was working on. And by looking at that, I said, look, if I'm able to find a compound here that is able to reverse my phenotype or aggravate it, and if I'm able to identify the mechanism of action, the target of these drugs, I should be able to quickly pinpoint new entities, new targets that will feed the traditional drug discovery pipeline, and at the same time, I will be able to use these targets, these probably unsuspected targets, to better understand the disease I'm working on, which I call chemical genetics, using these compounds to quickly identify targets through another approach, something that is not really used in the laboratory right now, and using then these targets to understand your disease. It's similar to a forward genetic screen, but using chemicals. So let me show you an example of that. So when I started my PhD, I worked on different models, including one which, in sea lions, a model of Duchenne muscular dystrophy. So it's nothing to do with the brain, but it's a muscular disease, and it's a very good example. So this model was showing a phenotype very similar to humans. So when you take out the gene dystrophin in the worm, which is the gene responsible for this disease, a muscular dystrophy, right? So when you take out this gene, the worm starts to have a progressive muscular degeneration, as in human, and a progressive loss of motor functions. And interestingly, this worm is perfect for establishing screen and testing drugs. Why? Because he has a defined number of muscle cells, 95 muscle cells. You can see here 4 muscle cells, which are diamond-shaped muscle cells in this worm. Okay, so they do not fuse, and they are in a number 95. So that means that, and they cannot regenerate. So when they degenerate, you will see them like hole like this in the muscles, and you can directly count this number of absent muscle cells which will be the perfect reflect of the progression of the disease. For example, this one has a score of 0, it's just healthy, and this one has a score of 4. And this number allows you to compare different treatment. So that was a very good model to start with, and great to establish screens, but you might understand that this is not really compatible with large-scale screen, and we had to engineer this to make it compatible with what we do in industry and to try to after sell that to the industry. So let's, for example, to quantify these muscle cells, you need to collect this animal, to wash them, to do a permeabilization of the cuticle, to do a phalloidine staining, which is actually quite expensive, by the way, in a large number. And after put this worm under slide and each population, and then count manually each worm under a microscope. So that's feasible, but it's not really compatible. That's feasible for a small pool of compounds, but not compatible with a large pool of compounds. So we had to find a way to make it more automatic. So what we did, we expressed GFP in the nuclei of the muscle cells, and as these muscle cells do not fuse as our muscle cells, they remain, they only have one nucleus. So then you can see in this worm that we obtain around 95 nuclei fluorescent dot, 95 fluorescent dot, reflecting the 95 muscle cells. So we had to do nothing but just look at this worm in a dish to evaluate the number of muscle cells in this worm, to qualitatively assess the musculature of the worm. You can see here absence of fluorescent dot reflecting muscular degeneration. But we went further than that, and we used a cell sorter, a kind of a cell sorter for worm, that was able here to evaluate these fluorescent dots and give numerical values that you can use to automatically compare your different treatments. So by establishing such a screen, we tested around 10,000 compounds. So we found a number of hits, and we validated 26 of them in mouse model. So first, that was a big demonstration. Yes, you can use small animal models to find drugs that come back and that present an alternative to the traditional drug discovery pipeline. Okay, that's very powerful. But at the same time, so I wanted to show that that could be also very useful more in terms of basic research, and that we can use that to identify unsuspected targets in our disease. So, but unfortunately here, I was surprised because I was a bit fooled. Understanding the function of even one drug can take longer than a PhD. It can be very complicated when you have no clue about how your drug works. So that was a big deal. But let's wait. Actually, so I told you that here we obtained 26 hits, and actually a bit more. But when we looked carefully at these hits, we found that there were only 3 main chemicals families, 3 main chemical scaffold in this pool, and that were belonging to sulfonamide compound, tricyclic compound, and benzodiazepine compound. And we know that there is approved drugs in the market belonging to this scaffold, and with known mechanisms of actions, we know few targets of these drugs. So let's take the example of sulfonamide compound. We know that they are strong inhibitors of the enzyme carbonic anhydrase. And there is, for example, 2 known drugs available in the market called naptazane for metazolamide and daranil for dichlorphenamide. So we tested these drugs in the worm, and as no surprise, they worked quite well. So you have here wild type with this 4 muscle cell, here the DMD with this absent muscle cell. So you can see here that this animal presents 6 absent muscle cells per side of worm. And when you treat this animal with dichlorphenamide, daranil, you nearly blocked completely muscular degeneration, and you also restore motor function. Only 1 absent muscle cell in this worm. And then, as we know that they are inhibitor of carbonic anhydrase, I looked into the worm to the available carbonic anhydrase. First, as the siligans present some carbonic anhydrase, and yes, we find 6 different. So we test them individually in the worm, and we found that actually downregulation of CEH4 was sufficient to block or slow down muscular degeneration. And we showed later that actually CEH4 was indeed the target of dichlorphenamide, and they were acting synergistically in reduction of muscular degeneration. So very powerful, right? So we find a really unsuspected target here involved in muscular degeneration that would probably not be found easily through another approach. But we went further than that, meaning when I looked at the expression of these different carbonic anhydrase, I realized and I saw that demonstrated that CH4 was the only carbonic anhydrase expressed in the mitochondria of muscles. And this helped me to really decipher and to point out my research to these specific organelles. And this helped me to find additional compounds that block muscular degeneration that were not identified during the screens, and additional targets, player of muscular degeneration, additional targets that we can use to try to treat patients. So briefly, we went with a model. I will not detail that. It's not the point today. But just to show you that chemical genetics approach, a drug discovery approach, allowed me to identify all new mechanisms in muscular degeneration and new drugs and new targets that involve mitochondrial stress, mitochondrial fission, and release of apoptotic factor, and so on. So that was very, very powerful. And just also to illustrate you the power of chemical genetics, when I started this research, this PhD, my pure wish was really to demonstrate it was feasible, and after to sell that as a kind of a technical approach, a platform to the industry. But when after his PhD, I really fall in love with basic research, which I mostly do today, and I wanted then to try to make a career in the academic. But my supervisor at that time was a bit stressed out because he said, I'm not sure it's a good idea because the drug discovery approach will never lead to good papers. I guess he's kind of right, but if you use this wise approach, if you use specific chemicals, as I will show you later, actually it can be very profitable for someone that do basic research and by doing chemical genetics. And just to illustrate you for DMD, just for this PhD, we obtained, related to this PhD, we obtained 3 patents and 5 papers starting from compounds and describing new mechanisms. So it can be profitable. So today,